Has lots of great things to tell us. So it is my pleasure to introduce Josh, who is a water quality specialist at the Niagara Peninsula Conservation Authority, and he's also a Brock graduate. He has 2 degrees from Brock. Right? So it's great to have Josh back to his campus, as we say, and it's great to have you talk in our seminar. Thank you, Josh.
Yes, you're welcome. Thank you for having me. It's a little bit weird coming back and doing a talk because I'm used to being the student 20 years ago and listening to people speak. So yeah, it's kind of almost surreal. And it's unfortunate, it's 20 years ago and I'm that old now. So I'm going to be giving you guys a presentation about what I do. And so I've called my presentation Tracking Watershed Health in Niagara. And this is basically my main job at the Conservation Authority. So for a presentation outline, everything you need to know about me in one slide. And then I'm going to do a little bit of background about the conservation authorities and then the water quality monitoring program. And then I'll go into a lot of detail about our water quality monitoring program at the Conservation Authority. So this is everything about me in one slide. And a part of it's already been mentioned, but back in 1993, I started at Brock University, and I did the Urban and Environmental Studies program in biology. That was my undergrad. And I think Dave Brown, he's still here, I believe, and he was one of my first-year professors. So a lot of the professors are gone now, retired. So in 97, I left Brock, and I ended up at Niagara College. I did a postgraduate certificate there in ecological restoration. And then a couple years later, I thought I'd return to Brock because I missed school so much. And I did my master's degree in biology. And I spent a lot of time doing research on Wainfleet Bog down in the southern tier there. I think for every weekend for 2 years I was in that bog. Did not get Lyme disease, so that's a good thing. There was a Lyme disease in Wainfleet Bog, which I found out afterwards. Ticks can carrying that. In terms of work, I hooked up with the City of St. Catharines as environmental technician assistant, so I was doing some of their pollution control work there. And then later on, I got hired on at the Toronto Zoo. I was doing restoration ecology. I was a restoration ecologist there. And then for 3 years, I spent at Agriculture and Agri-Food Canada. I loved that job. I was an entomology technician. We were doing work on biocontrols of pest insects in the greenhouse industry. Great program they had there. And it was sort of a up-and-coming research with these new predatory beetles. It was pretty cool. If you're a bug nerd like me, that was a great job. Unfortunately, that came to an end. That program only lasted 3 years. And at the very end, I managed to get snagged by the Niagara Peninsula Conservation Authority. And I've been there for 15 years this year, so in the water quality monitoring. So I've spent most of my career at the Conservation Authority. For most of it, I've actually been out in the creeks sampling away. And more recently, I'm now kind of sitting as a desk job doing a lot of the analysis, as my colleague had left a few years ago. So that's kind of me in one slide. I was going to do another slide about my personal life, but that's not that exciting. But, in terms of conservation authorities, when I remember starting the Conservation Authority, and even when I was in school, what is a conservation authority? There's so many governmental agencies out there. It's actually quite confusing. We have, the Ministry of the Environment, which is now the Ministry of the Environment and Climate Change. We've got the Ministry of Natural Resources and Forestry. We got Environment Canada. We have the municipalities. We have the conservation authorities. Like, where do they all fit in this kind of bigger picture of environmental and governmental agencies? Well, conservation authorities came about in 1946. There was the provincial government and the municipal governments got together and they created the Conservation Authorities Act. And that was basically in response to soil erosion and flood control. It provided means for the province and the municipalities to work together to form a conservation authority if they wanted and undertake programs of natural resource management. On a watershed basis. So watershed is the key word in that. We're set up by watersheds. Broad responsibilities depending on the conservation authority and their capacity. Watershed management obviously is the main responsibility, but there's a lot of community-based projects and municipal partners. So it's kind of a municipal provincial agency. We're actually funded from the municipalities, but the legislation comes from the province. It's a little bit weird that way. In fact, most of our board are municipal councillors, regional councillors from just over across the street, in fact. So, conservation authorities, there's 36 in Ontario. We're Niagara right there, but most of them are in southern Ontario, the east and the west. There are actually a 4 or 5 sitting up to the north. Every conservation authority is resource different, depending on what municipality you're in. Toronto, for example, as expected, has lots of people. There's lots of money there. They're the biggest conservation authority. They have most staff. They have the most programs. So they're really well resourced. And then you can go to the credit, which is Mississauga, another big municipality with Peel region. Grand River is another big conservation authority. Halton is also a very big one. So those are, I would say, the large ones with the biggest types of programming. We're a mid-sized conservation authority, and then there's very small ones. These northern ones are often very small and under-resources, 15 staff or 10 staff for the whole authority. So the programs they offer, as I said, everybody's resourced differently in their capacity, how much money they have. The big thing conservation authorities do, flood forecasting and warning. That's almost mandated in that we have to do that. Protect human life, protect property. That's the main core program at the Conservation Authority. But there's a whole slew of other things that have popped up over the years that we do. Watershed plans, watershed monitoring, what I'm doing, stewardship, septic inspections, fishery management plans, tree planting programs, conservation education. So when we have ownership of lands, we have parks for recreation. So there's a whole slew of programming that the conservation authorities provide. So this is our conservation authority, or my conservation authority. I don't know how many of you live in Niagara, but this is the Niagara Peninsula Conservation Authority watershed. It's 2,424 square kilometers. It was 1959. It was when it was founded. It is actually quite a unique ecosystem. We have the Niagara Escarpment kind of running through it. We have 2 Great Lakes, Lake Ontario, Lake Erie, the Niagara River. Our biggest watershed is the Welland River watershed, which runs from west to east, starting up here in Ancaster at the Hamilton Airport, Mount Hope area, and going through the watershed like easterly and draining historically into the Niagara River, and it encompasses a very large section of our watershed, and that is most of my attention, in fact. Another watershed we're well known for is 20 Mile Creek. That's the water course that goes over Balls Falls, one of our flagship conservation areas. And we have 12 Mile Creek, which is one of the highlights of Niagara in terms of water quality. So it keeps us busy in terms of municipalities. We have all of the Niagara region in our municipality, and we have a portion of Hamilton and Haldimand. And so those are our main funders for the programs of the Conservation Authority. If I can just say too, it's one of the most engineered watersheds in Niagara, or in Ontario. People from outside of Niagara don't realize how messed up we've changed Niagara's water courses. We put a canal through it, so there's like another channel that literally runs through the peninsula. And to do that, they've severed water courses and severed watersheds. We've also, in order to harness the power of Niagara Falls, we've created the OPG, the Ontario Power Generation, for electricity. So in order to do that, we have the Queenston Reservoir right there. In order to fill that reservoir, we actually redirect water from the Niagara River back down the Niagara River, or the Welland River. And so what that causes is a backflow of water all the way up to Wellandport here. The Welland River, in fact, never ever drains in the Niagara River. It actually goes down this power canal that feeds this reservoir in Niagara Falls. And so that causes all types of problems, and that also causes problems for the Welland River. It also has to go under the canals that are also going north to south. So our really highly engineered watersheds that are make this watershed quite interesting from just an engineering standpoint. I'm going to kind of, this is before I started the Conservation Authority, but I was quite aware of what was going on when I was in school. 1990s weren't very good times. I call it dark times for environmental monitoring. In Ontario, there was a recession going on in the 90s, and so guess what? Government slashed a lot of the environmental monitoring programs that we had. So it was awful. Our Conservation Authority had about 35 staff, and from what staff had told me, when this funding arrangement changed, they walked out 7 people that day. Because they had to. They had no more money. So that was a significant portion of staff just leaving. And that was all conservation authorities, Ministry of Natural Resources and Ministry of Environment. So that program cut put a big hole in our environmental monitoring in Ontario. See if I can get this thing to play. I know some of you know what this is. Now this is kind of a, a breaking point in Ontario, and people who've already seen it, I know Kate's seen it. Does anybody know what this is at all?
Is it a tunnel?
Okay, it's kind of, yeah.
Oh, okay. Well, I was gonna wonder if it was the tunnel that was made by Big Becky, but
Nope.
okay.
I had one person guess it was a colonoscopy, but it isn't a colonoscopy. So what this is an actual groundwater well, and it's the municipal supply well in Walkerton. And what you're seeing is a camera that's being scoped to go to this well and see what's going on. And you can see all this cloudy water entering the well. So this is surface water entering the groundwater through the casing. And so this is really the breaking point in Ontario where things started to turn around in terms of environmental monitoring and our thoughts about source water protection. So that gets us to the Walkerton well, the disaster. It really was a disaster. We live in Ontario. We shouldn't have the disaster that happened in Walkerton. It was in 2000. What went wrong? Well, we had a well right here. This is one of the supply wells that was in Walkerton. So Walkerton's in kind of southwestern Ontario. We had a breach in the well casing 2 meters below surface. So you saw that breach you saw all the silty water coming in. That's water coming in off the farmers' fields that's over here. So in 2000 in spring, we had a big giant rain event, kind of like we did yesterday. A big rain event came through, and all the water from this farm field happens to drain towards the well. The well, guess what? It was actually built in a swamp in 1965. And so when they tested that well upon construction, it tested positive for bacteria. So it already had bacteria in it when they tested it, and it required regular chlorination. Well, that was not done for the 22 years it was in operation as a municipal source supply. It also required regular monitoring for turbidity. It was never done for 22 years. It also required that buffer zone from that agricultural land. It never had a proper buffer zone from the agricultural land. So it was vulnerable to runoff from those agricultural fields. And the operators had no training in Walkerton. So they were actually later found guilty of falsifying their records for 22 years. So it was really, really bad. And at the time, the MOE and the public utilities had no resources because the funding wasn't doing very well in the 90s to actually check in on these guys and see what was going on and meet the requirements of their certificate of approval. So this is really kind of the watershed moment for the Conservation Authority programming that we have now. In terms of what happened in Walkerton, and it's sad because it's actually getting out of people's minds. When I have summer students come in, they don't even know what Walkerton is. So Walkerton, that's not that long ago. It's only 16 years ago, but it was awful. There were 7 people who died, 27 people who had kidney failure. 2,321 people were sick. They think about 42% of the cases were actually unreported. Cost $155 million, the whole thing. But it resulted in the Walkerton inquiry. We had the Ontario Clean Water Act. Source water protection was rolled out after this. So the Walkerton disaster, although it's awful, was really key and crucial to establishing water quality monitoring programs that we have now. And if you do get a chance, there's a guy, his name's Bruce Davison. He lived in Walkerton during this time. He does a great presentation, and he taught, his neighbor was one of the people who died. He does a great overview of what happened at Walkerton, and it's really something to experience. Now, that kind of brings my arrival on the scenes. So in 2001, so about a year after the Walkerton disaster, our Conservation Authority then decides they want to have a dedicated water quality monitoring program. They had kind of a water quality monitoring program before me, but this was now directed funding to have, we're going to have staff who are going to be monitoring water quality on a regular basis, not kind of ad hoc, where staff are doing it kind of multiple jobs. This is dedicated staff. So our mandate was to assess the water quality in our watersheds, the ambient water quality in our watersheds. So it's groundwater and surface water. We also had a requirement, we have to report to the stakeholders. We just can't collect the data. We have to go out there and report on it. We have meetings like this, in fact. I'm reporting on data. That's what I'm going to be doing here later, showing you some of our data sets. But I have to report to the public and report to our stakeholders and partners. So that's kind of my mandate and what I'm required to do. Now this is basically my job now. So I spend most of my time going out in our watersheds and doing surface water quality assessments. Our program consists of two different things. We do chemical monitoring and we do biological monitoring. So chemical monitoring, I have about 75 monitoring stations around our watershed, and I'll show you where those are in a moment. And we do monthly sampling. So what we're doing is we're going out with our pickup truck and driving to our monitoring sites, collecting a water sample, and sending it off to a lab. So what that gives us is a quantitative measure of the contaminants that are in the water. So we have a concentration. We know there's this much phosphorus in the water. And the good thing is there's actually research out there that talks about guidelines and ecological thresholds that we can compare against. So we can see, are we over any kind of threshold? Are we under? And make some sort of assessment about our water quality. Now, the problem with chemical monitoring is it's very expensive. One set of my samples costs $235 for one site. Now, I have 75 sites. I visit them 8 times per year. So imagine that times $235. So it gets in the order of $100,000 worth of just lab analysis to actually run our program. And we usually go from March or April all the way to November, December, depending on the year. And this year we've been fortunate enough to get out in the winter time because it's been clear of ice. But there are some disadvantages. Chemical monitoring is a snapshot of what's going on in the watercourse. It's, you're taking a picture that moment in time. So you might say there was a spill event or something happened. If I'm not out there, I'm going to miss it. So that's a disadvantage to chemical monitoring. Another disadvantage is the toxicity. There might not be science out there that can give us information about what's toxic to certain organisms. We might know certain parameters are toxic, but we might not know about other parameters. There are data gaps in the toxicity of certain parameters, and we don't know how they affect all aquatic life forms. So that's sort of an issue too. The other component of our programming is biological monitoring, and we piggyback the two together. So biological monitoring is really looking at the organisms in the watercourse and using them as an indicator of the water quality. So we spend our time looking at the benthic macroinvertebrates. I know some of you guys came out with us yesterday to see how that was done, and we almost got flooded away by the crazy high rain conditions. But the benthic macroinvertebrates are the insects, the animals that the fish eat in the watercourse. And I'm going to show you a bunch of pictures of those as my presentation goes on. You could look at fish as well. Biological monitoring can include fish. We don't do fish. It's more expensive and you need more intense equipment, electrofishing gear and all that type of stuff. And some people are using algae as indicators and bacteria as biological indicators. But we and most other conservation authorities actually spend time for biological monitoring using macroinvertebrates. Now, these animals are easy to sample. You can see some of our equipment there. There's my colleague Jake kicking the creek over here, and that's how you would grab those animals. So they're relatively easy to collect. It is intense in terms of sorting and identifying the samples, but overall their abundance, their diversity, and their sensitivity, because that's what the key thing, how sensitive are these animals to the pollution, give us an idea about the overall health of the watercourse. So the problem with the data that we get from benthics is it's highly variable, really all over, lots and lots of noise in our data set. So it's sometimes hard to detect fine trends. Sometimes it's not clear when you out your sample what's causing the problem, because sometimes your benthic critters, they don't tell you what parameters actually knocking or changing the community that we have there. So that's kind of a disadvantage, and that's why you piggyback the two in hopes that you can kind of look at it at two different angles to get an idea as to what is impairing our water courses. And of course, I mentioned it's very labor intensive and dangerous like it was yesterday. Excuse me for one moment. So our water quality monitoring stations. So for a conservation authority our size, we actually have very, very good coverage. This is 75 water quality monitoring stations throughout our watershed. And you can see that's pretty extensive. We have pretty fine resolution in some of our watersheds, but that's our network. We've had that in place up to about 75 stations for almost 10 years. So there's a lot of data that goes with those stations too. So we're trying to get to long-term data sets. In terms of key partnerships, I really want to highlight the partnerships because we're not doing it on our own as a conservation authority. We require partnerships in order for these programs to survive. So what happens at different political levels affects us. So Niagara region, they're great. They provide us $14,000 in in-kind laboratory analysis just across the street here. That's where it's done in the environmental lab. They basically said submit 10 samples to us a year, which is 80 samples actually overall, and we'll run the analysis. They get the data, we get the data. Similar arrangement for City of Hamilton. They give us $16,000 in in-kind sampling. The Ministry of Environment gives us $35,000. That's for the Provincial Water Quality Monitoring Network partnership that all conservation authorities have, but that's our cut of the pie. And then we have the Niagara River Remedial Action Plan, which is part of a federal government Great Lakes Sustainability Fund. They gave us up to $50,000 a year to do lab analysis. So this is all our lab analysis in kind and support that we get from our partners. And then, of course, we have our own money that we use to cover off some of the costs for our lab analysis. So what do we look at in terms of the water chemistry? Well, water chemistry, we generally look at nutrients, metals, bacteria, and a general chemistry. And that's, like I said, about $235 a sample. We do zero in on about some key parameters when I'm doing my analysis. Chloride. So we can, the source is a road salt and impacts. There's toxicity with having high chloride. Phosphorus, right? It's a nutrient for fertilizers, sewage. Nitrate, another nutrient. Suspended solids, so that's like from erosion. We have sediment entering water courses. E. coli, the big one, of course, sewage and manure, and then a whole series of metals that can be indicators of effluent and pesticides and stormwater runoff. And all these are very sensitive to urban and environmental land uses. So and they all have water quality guidelines that we can use. So that's kind of why we zone in on those 8. And there's a lot of information and science about these types of things. And these are the documents that when I refer to water quality standards and guidelines, these are your books that I spend a lot of time in. Water quality, this is the provincial water quality objectives, and this is the in federal government's water quality guidelines, and then that's the drinking water standard. So these things I use all the time to assess our data. So the next series of slides, I'm going to show you some of our data. So, because that's where I spend most of my time. That's what you guys like. You guys are in your master's program right now. You guys are going to be probably crunching some numbers down the road, right? So I spend time. I have lots and lots and lots of data. I have, like I said, 75 stations, 8 samples. How many parameters do I have? Probably about 25, 30 parameters that I'm looking at. So I'm going to zero in on 8, and I like to look at data in terms of temporal and spatial. So it's an easy way to show people our data set. So this is a kind of a spatial, distribution of our water quality data, looking at the median concentrations of chloride. So that was like the road salt possibilities. And this data set is 2010 to 2015. I took about a 5-year chunk here. And so the colors kind of give you an idea as to what the concentrations are. So the green are your lowest concentrations, under 25. And I'm not going to go through what the guidelines are, but I will say generally, in most of these figures I show you, if you see red or black, red is over the guideline, or at least approaching it, and black's usually really bad in terms of it's really high, in terms of certain concentrations. And like the yellow would be intermediate. So you can see, when you plot it this way, spatially, you can kind of see how you can start to identify some hot spots. So right away, you see some black areas standing out in these watersheds. So right away, there's one here up in Upper Welland River. That got our attention because the values were so high. Well, when we go out to that site and we explore a little further, well, that's Hamilton Airport. And there's a parking lot right there. And guess what? On their airport, they have a large salt storage dome. Well, guess what happens when it rains and when the water drains? So it goes right into that creek that we're monitoring. So that is why we get really high values there. And we've talked to them about that. And guess what? They moved their salt storage. So I'm hoping to see that number come down over time. A couple other sites here, chloride, 40 Mile Creek in Grimsby, right by the QEW, much like 30 Mile Creek. So it's probably some effect from the actual highway, because all the highway runoff goes right into those creeks. And there's a salt storage area right there beside 40 Mile Creek. So you're able to start identifying areas in the watershed that have these kind of hot spots, and then explain why they're occurring. And hopefully we can actually work out why, or possible ways to fix things. Some things are just kind of almost naturally occurring. We have a hot spot down here in Eagle Marsh Drain, which is down in kind of Wainfleet, Port Colborne. Well, the quarry there is pumping groundwater to support base flow for the water course there. So you're actually getting chloride from the groundwater. So base flow is doing really well, but you got a high chloride value there. Copper concentrations parameter. Generally, copper is not an issue in Niagara, but we have one hot spot there and a couple in there. We're actually going to do some investigation this coming year. There's some greenhouses out there, and we're wondering if that might be the source of them. E. coli. So, of course, I was talking about the red. The red and black are usually what I'm trying to draw your attention to. There's a lot of red, and there's some black on here. If I can point out one, which we've actually done work on this past fall, 2 Mile Creek here in Niagara Lake is actually off the charts for E. coli concentrations relative to the much larger data set. We went out to the Niagara Lake and did some actual investigations, sampling stormwater outfalls along the water course. So this is water that drains from the neighborhoods and goes through the sewer system there. This is stormwater. And it enters the water course. Well, we got really high values of E. coli out of those outfalls. I think one was 1 million. And so it's raising the water level concentrations of E. coli in 2 Mile Creek. And so we're hoping to do a little bit more of that. And we're working with the township, and they're trying to source track that down. Lead. Well, lead's not a big issue in Niagara. It's all under the guidelines, and we have really no lead issues. And that's probably my best map. Nitrate, another nutrient. Generally not too bad. For some reason, 12 Mile Creek, which is, I would say, our best water courses, has some high nitrate levels. Well, that one site there, that black dot, it's literally a first order stream, and upgrading of that is a golf course. So it's probably coming from the golf course. So we're hopefully working with the golf course to get that taken care of. Total phosphorus, a lot of phosphorus in our watershed. Our watershed is agricultural mainly, so we have corn, soybean, wheat, all up in this area. So there's a lot of phosphorus going down. We also have tender fruits and vineyards all through here as well. So there's a lot of phosphorus in our water courses. Total suspended solids is sediment. Couple spots that are hot. Oswego Creek, for some reason, in the Welland River watershed, has a lot of sediment entering the water courses, so we're going to have to take a look at that. Zinc, another metal. Again, Hamilton Airport up there, airport. They have galvanized roofing material. We think it's coming off that, and the airplanes, the brakes. The braking components actually apparently have zinc in them, and when they land their planes, they got to put the brakes on or they go off the runway. And so the brake material is washing off that and finding its way into the water course. That's as we're being suspected, at least. That's according to them. So we're going to see what's going on with that. So that's kind of looking parameter by parameter, and you can do that. And that is a good way of looking at our data, and that's a spatial way. I often have to bring all that data together. So how do I do that? And thankfully, there's a tool out there. And the federal government, Environment Canada, has something called the Canadian Water Quality Index. So you take those parameters, all those 8 parameters I just presented to you guys, and throw them in this cauldron, and it will spit out the Canadian Water Quality Index. It's a great tool if you ever start to do any water quality analysis. So what it does is it looks at the number of parameters that are not exceeding their that are exceeding their guidelines. Or are any of these parameters exceeding those guidelines? It also looks at how many times those parameters are exceeding that guideline. And then finally, it looks at the magnitude of how much that parameter is exceeded by. So if you really blow past the guideline, that's going to affect your rating more so than if you just kind of go over. So it accounts for all these 3 different things, and it's a great tool. A lot of conservation authorities use it, other agencies use it. It really stands up to scientific rigor. And you can use it at a local, regional, and even a national scale. So it's a great tool. In terms of how we roll this up in our data set, so I took those 8 parameters that have guidelines, and I threw them in the cauldron, which is just an Excel spreadsheet. And this is what they look like. So basically what it spits out is a water quality rating: poor, marginal, good, fair, and excellent. So those are your water quality values. So it kind of gives you an easy to digest rating that you can present to the public about what your water quality data is showing. And so when I do that, this is what our watershed looks like. About, I think the number, 60% of our sites are poor. So there's a lot of red here. So that's not unexpected when you saw some of those other figures earlier. We have certain parameters that are really high, and that's what's driving these water quality ratings. We have about 32% that are rated as marginal, 5% that are rated as fair, so those are the green ones, and we have 2%, 3%, sorry, it changed, that are rated as good. Now, those good ones, we'll talk about those real quickly. They're kind of artificial. One of those sites is the Welland Canal, so we actually take a sample out of the Welland Canal. Well, the Welland Canal is Lake Erie water. Lake Erie water is pretty good, like for the most part. That's our drinking water around here. So Lake Erie water is not blowing through. It's not subjected to a lot of the agricultural issues. There's so much water coming through, it's diluting everything. So that's why that rating is such a good rating. And then we have 12 Mile Creek here in Port Dalhousie. What happened again is through drinking water and through hydrogeneration, they redirect the Welland Canal water, which is Lake Erie water. Just over here, Lake Gibson, Lake Moody, falls behind Brock University and enters 12 Mile Creek. So you have this mass amount of clean water coming through, and it's artificially raising that value to good water quality. So I kind of put a little asterisk behind those ones when I'm trying to explain, it's not like that's some protected area. It's being diluted by water from Lake Erie for the most part. But that's kind of what our watershed looks like. It's generally a poor situation. So what are the main culprits? When I look at our data set, and I think you kind of got a clue when I showed you back a few slides in the individual parameters. Total phosphorus and E. coli are the two driving parameters that reduce our water quality ratings in Niagara. This is what summertime looks like in our watersheds in most of our Welland River area, which is the largest watershed. We have algae and duckweed. It looks like you can walk across the water. There's so much of that stuff because the nutrient levels are so high in those watersheds that there's an overgrowth of algae and duckweed. Of course, then E. coli. We have lots of E. coli in our watersheds that are causing issues and are dropping our ratings. In terms of what the phosphorus is doing, we actually had an opportunity to deploy dissolved oxygen loggers from 2008, 2010. We still actually deploy them to track summertime dissolved oxygen concentrations in our watersheds. And we actually found that this circled area in the summertime acted like a dead zone. There was actually very, very low dissolved oxygen. So this is the actual data. So this is concentrations of dissolved oxygen from May to September. That's your provincial water quality objective, which is about 4 milligrams per liter, or milligrams per liter. And that's the concentration of dissolved oxygen. And so what you get in these systems are sustained low or very low dissolved oxygen, so it's not coming above that. You do see that some that go up, and what you're seeing is a very daily diurnal cycle. You have it go up really high during the day because photosynthesis, and then it crashes at night during respiration as the algae and the duckweed kind of go to these extremes. And this is very, very difficult for aquatic life to actually deal with. In fact, the M&R biologist, Ann Yagi, had fish data, walleye data, and she was tracking where they go in the summertime. And guess what? They don't even go to that dead zone area. They're trying to scatter from that area. They're trying to get out of that dead zone area. So the fisheries data is matching what we're seeing with our dissolved oxygen logger data. So that's another way to kind of look at data, and that's kind of what phosphorus is doing to our water courses. Now the other component I want to talk about is the biological monitoring. So this is kind of what I did with a couple of you guys yesterday, and I love this. This is kind of, I've been a bug nerd since I've been a little kid. I used to play with ants and all that kind of stuff when I was a little kid. So working with bugs for a career is really, and this is kind of what I got hired for because I had so much background in entomology before I started. They wanted me to kind of be the bug guy. I'm kind of sad actually, my technician does most of the bug work now, but I still get my hand in it. But biological monitoring is looking at the animals, the insects, and the critters in the water that the fish eat. Now, I know yesterday we were a little confused about what the organisms were, so I'm going to go through exactly kind of what we were looking at in terms of the animals, just really quickly to kind of give you an idea. So it's not fish. These are like the insects. So this is a type of insect, stonefly. One of my favorites. These are very sensitive animals. Many different types. You actually find these guys in your water course. You should be very happy. Another good one to find are mayflies. Caddisflies. They build cases, most of them. This one actually, if I had it turned around, it would look like a snail shell. So it's a really specialized case. They're very interesting critters. If you find those three organisms, you find the caddisfly, the mayfly, and the stonefly, if you're doing a quick assessment, your water course is in probably pretty good shape. Some other animals we find, damselflies, dragonflies, more people are familiar with these. The dobson flies, these are big giant like fish flies or alder flies, common around here as well. Beetles, there's lots and lots of beetles in our water courses. True flies, so the diptera. So this is one of your probably your enemies, is the black fly. We also find like that's a crane fly, the chironomids or the bloodworms. Some of these are much more tolerant to pollution, and almost this is almost a stereotypical indicator of bad water quality. If you only find bloodworms, your water quality is not doing very well, and it's because these bloodworms are able to store oxygen in their hemoglobin. They actually create hemoglobin, so that's pretty neat that they're able to do that, but that means they can survive low dissolved oxygen conditions or poor water quality conditions. Crustaceans. I was hoping we would have found one of these yesterday in our field trip, but that's the crayfish. This is an amphipod or a side swimmer, an isopod. Some people have these in their gardens, but this is the aquatic form, so the other one's like a sow bug or a potato bug, people call them. They're roly-polies. So that's the aquatic one. They don't roll up quite the same. Mollusks, we find snails, we find clams and mussels. Of course, the worms, some of the least favorite of what people, when I go sampling, would freak out about. The leeches, very large leeches sometimes that we see out there, kind of going through the water. And of course, smaller aquatic worms. Very tolerant, these animals. They love low dissolved oxygen. In terms of collecting samples, there was that one photo I showed you of my colleague with the kick net. We predominantly use a T-sampler or Cerber sampler. Has a certain mesh size to capture those animals, and you just insert it into the creek, and you insert your arm down there, and you kind of disturb that creek. And what you do is you set it up so the flow goes this way, and so you're using that flow of the water to push those animals down the sleeve and into a collection vial at the very, very end. And so that's my colleague Andrea. She's out there collecting at the Hamilton Airport site. And there's good flow, pushing the water down the sleeve. You can see the sleeve extended. We take that little collection vial, we dump it out, and we sort through it, and we pick out the animals. This is the very labor-intensive part. And then we ID them and record their number. And so that's the process. So often we can only get to 3 sites a day, and with the replication involved, it takes us, another whole day to sort through the samples. So it is quite involved. So what do we find when I look at a sample? Well, this is a typical sample of a community with impaired water quality. So if I saw this in my sample after sorting through it, this is a lung snail, lots of aquatic worms. We have some chironomids or bloodworms. This is one of my favorites, the rat-tailed maggot, nice common name. It's a syrphid or a hoverfly is the family level. But if we see these guys are very tolerant to pollution. They love low dissolved oxygen. If we're only finding these, it's probably something going on with the water quality at that site. This is what you kind of want to see when you go to do some benthic sampling. This is the most exciting, lots of diversity. The big 3 I talked about before, those stoneflies, the mayflies, and the caddisflies. That's a beetle there. This is a water penny. Of course, that dobson fly crayfish, an amphipod. This is the bloodworms, the chironomids. You're going to find those in healthy sites as well. You just won't only find them. You'll find them in a smaller abundance, right? And so you want to see a diverse site, lots of different taxa, tolerant to sensitive animals. So what do I do with all that data? So I have, again, a similar situation. I have abundance data. I have, a taxonomy, so I've identified them, so I know what they are. And then I have their sensitivity values. There's a lot of research out there that's looked at the sensitivity of some of these organisms. So I then take that data and then throw them in my cauldron, and there's tools out there. We use one tool is a BioMap Index. There's something called the Hilsenhoff Index. There's a whole slew of biometrics that you can use to assess your water quality. Percent insects in your sample, percent this, percents amphipods and isopods. So there's a whole series of things you can use. In this case, we're using the BioMap Index. So again, it takes all that number, or all those numbers, and crunches it into, just spits out an index value, which would rate the water as either impaired, sort of an intermediate level, or unimpaired. And I put it on my map. And so these are the same 75 water quality stations we had before, where we do our water chemistry. Guess what? The BioMap stuff generally tells the same story, which is great. So we actually have, the majority of the time, we have the same type of story. Unfortunately, 81% of these sites are impaired. 7% would be the intermediates, which would be, so impaired are the red, intermediate are the yellow, the green are unimpaired, so that's a smaller percentage, 5%. And 7%, the black ones, so the black are not really bad. They're only bad because you can't get into them, they're really dangerous. They're like, way over our head and flow, and so we don't sample those ones. So some sites are just unsampleable due to the conditions that are there. But generally, these match what we're seeing out in our water chemistry too. So it's a good way of looking at the data in two different ways, and they're telling the same story. There are some occasions, though, where we have water quality is unimpaired, but the benthic community is not there. So what's going on? So sometimes we have to look at, are there habitat things going on at that site? Because the habitat's a clear thing. If it's just a concrete channel with no habitat inside the creek, that's obviously an issue. So there's things to consider when there are kind of outliers like that come up. The other program I'm in charge of is our groundwater monitoring program. So this is kind of once a year I manage to go out and collect water quality sampling at our groundwater wells. So we have 15 wells as part of our network. They were put in 2003, and they're monitoring the key bedrock and overburden features in our watershed. We're actually expanding it. My colleague had an opportunity through the Ontario Geologic Survey to expand our monitoring of groundwater, and he's taken it on because I don't have any more time. And he's added 30 more groundwater monitoring stations to this already good network in terms of groundwater monitoring. So he's going to have his hands full because these guys keep me busy when they actually kind of amp up. So I'm keeping groundwater levels, track of that by deploying loggers inside the wells themselves, and we go out once a year and collect water samples. So that's what they look like, our wells. It's not like there's a pump on it, but that's kind of what they look like, and there's sometimes a box on it with some of our telemetry equipment so we can send data to our central databases, and then there's my colleague taking a water sample. So what is the groundwater data telling us? Provincial Groundwater Monitoring Network, that's that provincial program we're in partnership. Well, most of our water quality meets the guidelines. There are a couple instances where we have some exceedances, fluoride, boron, sodium. These are exceeding because of the natural conditions in the bedrock. So the water's been sitting there for a long time. It dissolves some of in the metals and the boron off the rock, and then it's found in concentration inside the water that we're pulling out of the ground for our samples. So that's naturally occurring groundwater issues. In fact, we're working to address that because that is obviously an issue. We have naturally occurring exceedances. Shouldn't really drink the water if it's over an exceedance. So how can we better get that information out to the public? We also had a couple of the shallow wells, either the overburden wells right into the soil, exceeded for nitrate. So nitrate past a certain concentration is known for causing blue baby syndrome. I don't know if you're familiar with that. That's when if you bottle feed your infant and use water that has high nitrate, basically the nitrate binds up the red blood cells, and your baby basically cannot absorb and get the oxygen. So it's not a good thing. So there's a couple wells that actually had really high concentrations of nitrate, but we actually did an investigation. We went out there and very localized to those wells, and it was because of the land use, agricultural right beside it. The other adjacent wells were not affected by that. Get it back up. So it gets to my reporting. So this is kind of my second part of my mandate. Like, what do I have to do with all this data? So every year I produce a water quality monitoring report. You can find that on our webpage. You can also, I do special, it depends on the target audience. Sometimes we do intense studies with other agencies, and so if you look up our well and river eutrophication study or our nutrient trackdown study, you'll find more intense analysis than you would find sort of in these water quality reports. This is more for public consumption. These are more for more technical consumption by experts in the field, but all available from us. One thing I want to highlight is what conservation authorities and our conservation authority do is it's a watershed report card. This is Conservation Ontario is kind of an agency overseeing all the conservation authorities, and it's encouraging conservation authorities to present all their data simultaneously using standardized criteria. So, and easy to understand criteria, like a report card. We're going to give A's, B's, C's, and D's for our watersheds. But how do you do that if everybody is using different standards to compare against? So there was a big, long process that went down by conservation authorities to try and standardize our reporting for these watershed report cards. So we did one in 2012, 2013 for our watershed. And in terms of water quality, what my expertise is, total phosphorus, E. coli, and the benthic data were used for the report card to calculate that. There was also forest conditions that were used. I wasn't involved in that, but our forest person looked at forest cover, percent interior forest, and percent riparian forest zone cover as indicators of forest health in our watershed. And there was also a groundwater component as well. But this is kind of how we used the watershed report card approach to give our grade. So in terms of water quality, if you had total phosphorus that was below that concentration, you had E. coli below that concentration, and benthic score using not the BioMap Index, but a different index called the Hilsenhoff Index, you grade your water course an A. And it would depend on where your water course fell in terms of this data set, and you would be able to grade your water course based on that. And of course, A to F means what we know it to mean, A being good, F being not so good. So this is our watershed when we put all our water quality data through the watershed report card, and it doesn't really deviate much from what we find in our regular watershed reporting. I would give our watershed a D overall. Some people like to say, what's our water quality like in Niagara? If a big, broad regional thing, I'd give it a D, and that's how it scores out. But I fine-tuned it. I just didn't give it a D. You can go actually zoom in and look into some of the specific watersheds, and some of the watersheds actually don't score too bad. We actually had some Bs and Cs as well. So it's not like it's the end of the world out there. There's a lot of work that needs to be done, but there are some nice areas in Niagara that have very good water quality that we need to protect and ensure that they don't change, and we have areas we need to improve. Now this is draft. I'm sure it's allowed to be filmed, but this is, I'm just kidding. I don't know, actually, you can film it. This is, I snuck this slide out of somebody else's slide, but this is the actual watershed report card data from all the conservation authorities. So when I talked about all the conservation authorities taking their watershed report card information and then putting it on a map, you can see this is what it would look like if you did that for all the conservation authorities when you take their data. So unfortunately, the colors are not the best, but generally we match what you would find in southwestern Ontario. We're highly agricultural, much like you would find down here. So our grades are similar to that. In some cases, better up towards the Bruce, and in the higher north you go. Some conservation authorities did not participate in this process. The Grand River, for example, unfortunately did not participate. A huge conservation authority, do all kinds of great stuff. They didn't seem to want to participate in the watershed report card process. So we're hoping the next one's coming up in a couple years, so we're hoping they'll be involved and present their data. But that's a neat way of kind of putting all the data, the report card data, into kind of a general release. And an A means an A in our watershed as it does the other, because everybody's using the same standard. So it gets to kind of my final thing. So that's all kind of the data. That's kind of the state of the watershed. I like to think of myself and our program as the doctors of the watershed. That we're kind of like, we administer the blood test to you guys, or the watersheds, and we get the data and we look at your blood test score. Oh, you have high cholesterol. You have high, low iron and stuff like that. And so we need to treat those things. So that's where this restoration programs at our conservation authority come in. So I'm telling our conservation authority or our restoration staff, stewardship staff, okay, we have some issues, and this is what the issues are. You have high phosphorus, you have E. coli, you have these issues. And so we have programs at our authority which are to work with landowners, and they're usually funding programs, cost-sharing programs called best management practices that focus on water quality improvement. And so manure storages. We're trying to encourage people who have large manure storage piles, contain it. Don't want to leave it out in the open, because that's going to end up in our water courses. So we'll fund, those are big expensive projects, $30,000, $40,000, $100,000 depending on the size of the operation that gets those. Wastewater containment. We've been working with greenhouses to contain their wastewater so they're not shooting it out into the watersheds, because that's really highly high in nutrients. It's water that's entering our water courses. It's very difficult for the watershed to kind of absorb that. It just kind of creates chaos, especially if it's released in the summertime. Riparian buffers. We need buffers between our water courses and our agricultural operations. So water doesn't run right off the agricultural lands directly into the water course. It's filtered by these buffers. So we work with landowners to put buffers in. Erosion controls. We put in these special fascines and crib walls that protect the banks that have been eroded, and they do a recontour, and just to ensure that we're not losing sediment and property. And then access to livestock. We do livestock access control projects where we're funding landowners so they're not allowing their animals into the watercourses. I also try and target specific watersheds for these stewardship projects. So this is an example of a watercourse or watershed. This is Beaver Creek, so well import area, kind of the central Welland River. Really, really high total phosphorus. It's total phosphorus concentrations. Beaver Creek, that's your water quality objective right there, which is around 30 or 0.03. You can see we have really, really high phosphorus in the Beaver Creek. So around 2009-10, we had the results of the Welland River eutrophication study. It recommended we target this watershed for nutrient best management practices or stewardship projects to contain or control the phosphorus that's in that watershed. Well, guess what? That targeted restoration approach is actually working. We're actually seeing, when I do trend analysis on this data, concentrations are decreasing. They're still, 600 and 500, which is really high when you want to be around 30, but it's still making a statistically significant decrease in the concentration. So there is actually a measurable amount that we're actually seeing in terms of our work. It's very difficult to show measurable progress in a short term with the stewardship programs in our watersheds. It's actually a very complex thing. You just can't put something in and assume you can fix it instantly. These are non-point source, so you can't really figure out exactly where the problem is coming from. It's not like it's a pipe spitting out water, an effluent where you can just turn off that pipe. You have this whole massive area in the watershed that's contributing, and each is contributing some part, and it's hard to kind of narrow that down. So this was actually one of our big success stories in terms of showing that we can control nutrients in a watercourse. Hopefully we can get it down lower. Sort of other actions that I'm trying to encourage is incorporating more data logging equipment. We're very much into spot testing with our lab analysis and stuff like that. But I want to find resources and funding to actually deploy more monitoring equipment, logging like dissolved oxygen, temperature, these big large parameters into the watercourse to actually track the different water quality parameters. And then we can provide that information to the public and also assist with our analysis. So that's a very key component in expansion that we're trying to move to. Another key thing we're trying to do is we're trying to be more aggressive in tracking down sources of water quality impairment. Much like the 2 Mile Creek, when I talked about the high E. coli levels we had, we aggressively went out there and we're out into our conservation area, going to these outfalls and sampling the outfalls in the summertime outside of our regular sampling runs to see what's going on so we can get more answers and point the other agencies to the problems. The problem is certain agencies are understaffed. They're doing the best job they possibly can, but they can't run around and do the kind of the investigation. So certain those hot spots, when I plotted all that data up and showed you some of these hot spots for certain parameters, we can go out there and sometimes see what's going on. And that 2 Mile Creek was a good success in terms of track down. That's my presentation and kind of an overview of our water quality monitoring program. And what the stewardship programs are doing, kind of what we're hoping to improve our watersheds, and what conservation authorities are doing, and our particular conservation authority too. So thank you for your attention. It's always fun and great to talk about our watershed and what our water quality monitoring program. So I will leave it at that. I hope I didn't go too long. But
Thank you, Judge.
yeah.